This slide is showing the cytoskeleton of the cell. So cells aren't just a blob that cannot contract. Sometimes they are if they're a fat cell, but muscle certainly is able to contract, and that's because of parts of the cytoskeleton. So there's three parts of the cytoskeleton. They are called microfilaments, intermediate filaments, and microtubules. So the microfilaments are extremely tiny molecules, and one example of them would be the actin molecule. And the microfilaments are responsible for things like cell motility, the movement of it, change in cell shape, and exocytosis and endocytosis. So for actin, it would be responsible for muscle contraction. And we're going to specifically talk a lot about it when we get to chapter 9. For change in cell shape, that would be muscle contraction, as well as endocytosis and exocytosis. Remember that endocytosis is when molecules enter the cell, and exocytosis is when molecules exit the cell. So these can kind of work like a roadway system in the case of endocytosis and exocytosis. The next example is an intermediate filament, and the intermediate filaments attach to desmosomes, which remember is a type of junction found in places like the cardiac muscle, as well as the skin, where stretching occurs. It's important for mechanical support and also protection of the cell from damage. The third example is a microtubule with a spherical protein that has subunits called tubulin. And these microtubules are going to be mostly responsible for things like centrioles. And pairs of centrioles make up what's called a centrosome. And they also help determine the shape of the cell, specifically in the case of mitosis, which we will be talking about soon. They also can act as tracks for motor proteins. So these motor proteins can act like feet to attach to these tracks and allow for a vesicle to walk from one end of the cell to another end of the cell. Next, we have a non-membranous organelle called the centrioles. You can see that they're containing microtubules, which what you just saw on the last slide. It was a type of the cytoskeleton. So the microtubule structure under a micrograph looks like what you see on the right-hand side of this slide. Those centrioles, when they are paired, there's two of them in what's called a centrosome matrix. So the centrosome is located near the nucleus, meaning cell center, and that is a microtubule organizing center consisting of granules. These granules are a pair of barrel-shaped microtubular organelles, and these microtubules radiate from the centrosome, and they're going to aid in cell division, specifically mitosis. Also, some of them are going to form the cytoskeletal track system that allows vesicles to move from one end of the cell to another. Centrioles also form the basis of what are called cilia and flagella. And cilia are in specialized regions of the body, like the trachea, for example. Both cilia and flagella are extensions from a cell. Cilia are stationary cells. They're whip-like modal extensions on surfaces of certain cells, like the respiratory cells. And the example would be in the trachea. And you'll learn about those specifically in chapter 4 on histology. So there's thousands of cilia. Their job is to act in a sweeping motion to move substances across the surface in one direction. And they move the substance of mucus. And in the case of the trachea, imagine that we're going to take this and rotate it 90 degrees so that it is running alongside your windpipe or your trachea. So if this is our trachea, the cilia are going to be sticking out like brooms, and they're going to be constantly in motion. The reason that they're constantly in motion is they're really part of your immune system to sweep mucus out of your lungs so that you don't get something like pneumonia. So they sweep it out of your lungs, then you can either spit it out or it can go down your esophagus and you can swallow it. The next example of this is flagella. But before we talk about flagella, I want to show you another example of cilia. So you can see that there's mucus on the surface. This is essentially the same thing that I showed you up here, just a much better drawing of it. So these cilia are going to be sticking out, and their job is to sweep the mucus across the cells. Flagella are longer extensions that propel a whole cell, so they would be like the tail of the sperm, and they're only in males. So only a part of the human population has flagella in them, but both of these structures, whether they are cilia or whether they are flagella, are made up of microtubules synthesized by centrioles. And they form the base of the cilium or the flagellum. Our next slide is showing the nucleus and the nucleolus. So there is a double membrane barrier that encloses the jelly-like fluid called the nucleoplasm. So instead of it being called cytoplasm, it's nucleoplasm. The outer layer is going to be continuous with the rough endoplasmic reticulum, and the inner layer is a network of proteins that maintains the nuclear shape and acts as scaffolding for DNA. The nuclear pores are proteins, just so remember all pores and channels are always proteins, and they are specialized to only allow for mRNA to exit the nucleus. The DNA is found only in the nucleus, and that's kind of like gold. So we have the nuclear pore shown here, the nucleus. So the nucleus is the control center of the cell. The nuclear envelope is the double-layered membrane, so it's like a phospholipid bilayer that surrounds the nucleus. And in this case, the DNA is in the form of chromatin. So it's condensed DNA. And at the beginning of our next video, we're going to talk about DNA and then about cell division.